Well, I'm starting off just to set the scene to some degree. And I'm starting here with a definition of synthetic biology, where we've just simply taken from the dictionary the definition of the word synthetic, where there's two broad meanings in common use. And one is that this synthetic might be related or involving synthesis, that is the construction of elements from individual separate devices or elements. Or it could mean that it's something that's prepared artificially or not of natural origin. And those two definitions, where you've got, in one case, synthetic elements which are constructed, or synthetic processes that are constructed, and those that are artificial, reflect the two extremes, if you like, on the diversity of synthetic biology, because you have various types of scientists who have different interests, either in creating artificial living systems, often from non-organic bases, from chemicals and things which might be on the shelf, at one hand, and on the other, you might have scientists who are interested in modifying existing systems to build new biological systems using existing parts. So you've got these two broad areas of endeavour which are being driven by the increased capacity to manipulate biological and biologically like systems. And of course, this idea that biological systems can be modified is something we ought to be familiar with. Perhaps we're not as familiar as we should be, in that biological systems, and I show here examples of plants, where the existing crop species which you're familiar with, that you'll see in your local supermarket for corn and tomato, are derived by many thousands of years of breeding from their antecedents, which these progenitors are essentially wild species that were domesticated by early hunter-gatherers. And in some cases, you have up to 10,000 years of manipulation of that plant species to produce modern varieties. So this idea of breeding to change the characters of both animals and plants is something we're broadly familiar with, if not comfortable with. However, there's now, with increasing technology, there's the prospect of being able to not just select and choose things with different characters over this long time span, but also over a much shorter time span to rebuild living systems, essentially using engineering principles and using our scientific base as a means of creating redirected patterns of growth inside living organisms. And so this prospect is now upon us in biology, but of course there's many fields where this has already been addressed. And I'll give you one quick example, which is something that came out of what used to be a quite esoteric topic, solid-state physics, which now underpins many of our daily activities. So here we have a picture of the first transistor. This mucky-looking thing here was invented or created or built in 1948, and it had the properties of an amplifier and a switch, and that was very rapidly adopted as a replacement for clumsy and expensive and energetically expensive thermionic valves. And so these transistors very quickly became commodities that were used in electronic circuits, and by the 50s, these are in wide use. By the end of the 50s, you now had integrated circuits, and this is actually a picture of the first integrated circuit, where you've got multiple semiconductor devices on a single chip. As time went on, you now have perfection of the construction techniques to build these chips. What's more, we have standardization of these construction principles. So not just the basic core functionality, but you have electronic, electrical, and mechanical interfaces which allow these components to be used by engineers. So you have engineers who might be building these circuits, you have engineers who might be integrating them into larger scale things, and then people who are making and selling machines, where these standard components mean that people can specialize at one level, and you have this abstraction which allows an industry to be formed around what was a very esoteric academic pursuit. And of course, with the progression of these standards, which is still in place, you now have today's microprocessors, which allow essentially many hundreds of millions of elements to be integrated onto single pieces of silicon. Now, this process of going from science to engineering is something which is upon us in biology. And the question is, how much will this apply to living systems, and what kind of consequences might that have? And of course, living systems are very different. Even something as complex as a modern microprocessor is nothing compared to the complexity that we find in these images, for example, of a growing plant, or in fact around us. The kind of complexity that you see in living systems is orders of magnitude more than any human artifact. If you look at your neighbour, you'll see that the brain behind that face, which might have 100 billion neurons working furiously at this point, is something which we can't even think to match. And yet this kind of complexity is built by cell processes and fed by glucose in a very cheap and economical way, and it self-organises. And this next image, which is taken from the BBC Natural History Unit, shows a plant, and I try and use this to represent the dynamic process of growth in a living system. So here we have a leaf with a stolon, which is coming from the tip of the leaf, and this small bulb of tissue at the end of this extrusion of the leaf continues to enlarge, and these processes are driven by growth. So cells are proliferating and differentiating in an ordered fashion and producing the container for a nepenthe, which is a pitcher plant. And you can see as this thing grows, it forms a hollow organ, you might say precisely engineered in a sense. With a lid, it's a hollow organ, the lid will now pop open and produce what we recognise as a pitcher plant. And by virtue of this time-lapse image, you see these processes compressed into something you can directly visualise. And so we know that these processes are driven by individual cells which are controlled in turn by DNA. And we have the underpinning technology which describes this process goes back to the early 50s with the structure of DNA being discovered, followed by technology from 1975 here, the invention of DNA sequencing techniques, which allows us to analyse now not just individual small pieces of DNA, but whole genomes, with 2001 giving us a sequencing of the entire human genome with its 3 by 10 to the 8 nucleotides. And we're now faced with, as of 2 years ago, the synthesis of the first genome. This is an entire genome, admittedly the smallest one we know, from this microbe, which is Mycoplasma genitalium, from the Venter Institute. And so we've moved through these series of slides essentially paralleling the kind of technological advances that you see in microelectronics. You have increases in capacity not just to analyze DNA sequences, but to build them. But the paradox here is that although we can build this has actually been built. It took probably $2 million to create. If you had $2 million, you could go and build something like this tomorrow. You could build your own genome. But the thing is that our capacity to design new genetic elements is many orders of magnitude less than the ability to copy an existing genome. And we're in a position now where we have this capacity to build, but not a capacity to design. And that's where synthetic biology resides at this point. And the way forward, I would argue, lies with the kind of ideas that were originated by Tom Knight at MIT, where he proposed the invention of standardized parts for biology, where you could take the kind of approach that engineers take in every other field, where you have standardized parts which allow the construction of larger systems in an abstracted fashion. And these biobricks, or small biological parts, are actually pieces of DNA which have defined function, which can be slotted together in a regular fashion. And I would try and represent the benefits of this in a simple way. So if you imagine that piece of DNA might encode a protein. In this case, it's a particular protein from jellyfish called green fluorescent protein. It's literally named. It's a protein that, as it's made, it folds itself up and converts itself into a fluorescent green molecule, widely used in modern biology. And so this corresponds to the DNA sequence within this modular piece. And it allows you to sort of abstract that DNA sequence. And these standardization approaches allow you to put different pieces together in a standardized way. So we now have essentially small tags, if you like, at each end of the gene, which allow easy assembly of larger scale DNAs. And the interesting process here is that if, for example, people working with the actual protein itself discover a variant, which has, in this case, say, a brighter color, you can make a part which is essentially interchangeable with the existing part. And this can be collected in a repository or a library, exchanged freely, and used to build subsequent devices. Within this interchangeability allows the decoupling of the design process from the fabrication details. And that's what happens in essentially every other type of human engineering, but now applied to biology.